everyone, thank you so much for clicking onto the video. Here are today's learning objectives. So the first is to be able to know that proteins are sequences of amino acids joined by peptide links, and then look at the importance of hydrogen bonding and sulfur-sulfur bonds in proteins. Then we're going to look at the primary, secondary and tertiary structure of proteins and the hydrolysis of the peptide link which produces the constituent amino acid. We're then going to look at thin layer chromatography and how amino acids can be located on a chromatogram using developing agents such as ninhydrin or ultraviolet light and identified by their RF values. So by the end we should be able to draw the structure of a peptide formed from up to 3 amino acids, draw the structure of the amino acids formed by hydrolysis of a peptide, identify primary, secondary and tertiary structures in diagrams, explain how these structures are maintained by hydrogen bonding and sulfur-sulfur bonds, and also calculate RF values from a chromatogram. Now I'm going to introduce proteins. So amino acids link together to form peptides, and a chain of 50 amino acids or less is called a polypeptide, whereas a chain of more than 50 amino acids is called a protein. And polypeptides and proteins are both polymers which are made by condensation polymerization. And what we mean by that is that a small molecule is liberated when the amino acids react together, and in this case it's water. And these polypeptides and proteins are also known as polyamides. We say that proteins are sequences of amino acids which are joined by peptide links. And naturally occurring proteins include wool, muscle, enzymes, and also hair. Each of the 20 naturally occurring amino acids are all given a non-systematic name, which is abbreviated to 3 letters. So what we mean by non-systematic is not the IUPAC name. So a particular protein will have a fixed sequence of amino acids in its chain. So for example, we have gly-allys-cis-val. So this particular protein is made up of glycine, alanine, cysteine, and valine, which are all amino acids. And this is actually the primary structure of a protein, and we're going to look at what that means a bit later. So at the bottom we have an example of protein, and this is just a small section of it, but it's actually a very, very long chain molecule. And what we have here, this CONH, is what we've learned as an amide link when we've looked at condensation polymerization. But in this specific case, where we're looking at proteins, we call this the peptide link. So the peptide link and the amide link is actually the same thing. It's just this C double bond O and then NH group. And this is what joins together all of the amino acids. So now I'm going to quickly remind you of how to form dipeptides. So when 2 amino acids join together, they form a dipeptide. And this is a condensation polymerization reaction, as we liberate a small molecule of water when the 2 amino acids are reacted together. So this water is a byproduct. And to join 2 amino acids, the amine group of one reacts with the carboxylic acid group of the other. And so because amino acids have both an amine group and a carboxylic acid group, we can use either 2 of the same amino acid to form a dipeptide or 2 different amino acids. And so in this example, we're going to use 2 different amino acids. So we're going to form glycis. And when we write it as glycis, that means we're reacting it in that order. So what we do is when we add these 2 together, we remove the water. So if I rewrite these as H2N, and then we have the carbon and the 2 hydrogens, and then a carboxylic acid group, and then we're adding it with this cysteine, so COOH, and then we have CH2, and then SH. So these amino acids will either be given to you in a data booklet or just in the question. And what we're doing when we go through condensation polymerization is we remove a water. So what we do is we remove this water here. And if I write this NH2 out, so we have N and then H2 like this, then the water we're removing is this one here. So we remove one of the hydrogens off the amine group, and then the OH off. And so this forms H2N, C, and then H2. Then we've got carbon, double bond, oxygen, and then we have a nitrogen bonded to a hydrogen, and then we have the CH, CH2SH, and then the carboxylic acid group on the other end. And so this is our glycis peptide, because we've reacted two amino acids together. And so what we have here is our peptide link, or our amide link. But in this particular case, we call it a peptide link. And a really important thing to note is that because this dipeptide still has this carboxylic acid group and also this amine group, that means we can react it with more amino acids to form tripeptides or tetrapeptides or polypeptides, so proteins. And so tripeptides means 3 amino acids reacting together, and tetrapeptides means 4 reacting together, and proteins are just lots and lots of amino acids reacting together. So the example we're going to look at is the formation of glycis ALA from glycis, which is a dipeptide we just formed, and then alanine. So here we have the structure of alanine. So first I'm going to draw out the glycis. So we have H2N, and then CH2, and then we have the C double bond O, N, H, and then CH, CH2, SH, and also the carboxylic acid group. So again, we do the same thing. Because we've written it as glycis ALA, that means we're reacting the alanine on the right-hand side, basically. So we write the alanine next to it. So we have H2N, and then CH, COOH, and then we have CH3. So we have our alanine. And again, we remove the water. So I'm going to write this as NH2 like this. So we can remove the water. So we remove the OH from the carboxylic acid and then a hydrogen off the amine group of the other amino acid, which in this case is the alanine. And so what we form, so this water comes out, and then what we form is H2N, CH2, C double bond O, N, H, CH, CH2, SH, and then now we have C double bond O, and then NH, and then CH, CH3, and then COOH. So this is our glycis ALA, and this is a tripeptide because we've reacted 3 of the amino acids together. And so here we have the peptide link, and also here as well. So basically what we have is that for every amino acid we have, so when we react 2 amino acids together, we form 1 peptide link between them. And so because we have 3 amino acids this time, we have to have 2 peptide links because we need 1 peptide link to link 2 of them together. And again, it's really important to note that this tripeptide still has this amine group and this carboxylic acid group, so it can react with more amino acid to form tetrapeptides or proteins. Now we're going to look at the hydrolysis of proteins. So this is very similar to when we were hydrolyzing condensation polymers. It's actually exactly the same thing, but just with longer chains. So proteins can undergo hydrolysis to form their component amino acids. And this hydrolysis is achieved by boiling the protein for 24 hours and also with 6 moles per decimeters cubed of hydrochloric acid. And we're going to see why a bit later. The protein breaks down to form a mixture of all the amino acids which make up the original protein or polypeptide. And all the peptide links are hydrolyzed by the acid, which just means that they're all broken. So in general, what we have is that first we have a polypeptide, or in this case, I'm going to draw a dipeptide. So we have H2N, and then C, an R group, a hydrogen, and then another carbon with a double bond oxygen, and then nitrogen and hydrogen. And then we have with an R group, and then a hydrogen, and then we have the C double bond O, OH. So this is our dipeptide. We know it's a dipeptide because we only have one peptide link here. And then we add some water with the presence of a H+ catalyst. So this H+ catalyst comes from the hydrochloric acid, and we form 2 of this amino acid. And in this example, we've assumed that we've used the same amino acid to form this dipeptide. So we have H2N, and then carbon, an R group, a hydrogen, and then the C double bond O, OH. And what we've done basically is we've just looked at this peptide bond, this peptide link, and split it down the middle. And then we've added the components of water back to the different ends to get this amino acid. So the mixture of amino acids can then be separated and identified using paper chromatography. And we're actually going to see later that we have a more accurate process, which is thin layer chromatography, which we'll be using to separate and identify the amino acids. And certain enzymes actually partially hydrolyze specific peptide bonds. So for example, trypsin, which is an enzyme, will only break the peptide bonds formed by lysine and arginine, which are both amino acids. So chemists can use this characteristic to find the sequences of amino acids in different proteins if certain enzymes will only partially hydrolyze some specific peptide bonds. So an example of a question you could get would be to write the equation for the hydrolysis of a certain polypeptide. So here we have a tripeptide, and so what we do first is identify all the peptide links. So we have one here. So remember that peptide link is the C double bond O and then the NH. And we also have one here. So these are both our peptide links. And the second step is to split them up. So we split them down the middle. And this gives us our 3 amino acids. Now, the final step is just to add the components of water back onto the different atoms. So on the carbon atom here, we need to add back an OH so we can reform that carboxylic acid group that we once had on it. And for the nitrogen here, we need to add a hydrogen, and then it's the same for the other peptide link. So what we get is we have H2N. So this is adding water and also with a H+ catalyst. So we get H2N, and we also need 2 waters actually because we're breaking 2 peptide links. And so we get H2N and then CH, CH2, CH2, 3, and then NH2. And then we have the C double bond O, OH. So this is our first amino acid that we form. And then we also have the nitrogen with the 2 hydrogens now because we've re-added the H plus from the water. And then we have CH3, and then we have C double bond O and OH because we've added the components of water back to this one as well, to this side also. And then for the last one, we just add the H plus back onto this nitrogen. And so you get NH2CH, and then we have CH2SH, and then this carboxylic acid group here. So these are all our amino acids that we form after the hydrolysis of this tripeptide. Now we're going to look at the bonding between amino acids. So the amino acids in a protein chain can bond together in a number of ways, and the first way we have is hydrogen bonding. And this is between the carbon double bond O group from the carboxylic acid of the amino acid and the NH group from the amine group. And it's really important for us to note that hydrogen bonding is a type of intermolecular force and not a type of bonding. So whenever questions ask you about the bonding between amino acids, make sure to mention that hydrogen bonding is actually an intermolecular force. The second type of bonding we have is ionic interactions. And this is between side groups or side chains of amino acids, so not the main bit of it. And so, for example, we have the CO- group on glutamic acid and NH3 plus on lysine. So if we have a look at the diagram on the right, if we first have a look at the hydrogen bonding, which was our number 1, what we have is the hydrogen bond between the oxygen of the carboxylic acid bit, the CO-, and then from the NH group, we have a hydrogen. And so the hydrogen and oxygen bond together because there's a very big electronegativity difference between oxygen and hydrogen because oxygen is much more electronegative. And so we form that hydrogen bond. And then if we look at the diagram on the left, there's electrostatic interactions or ionic interactions between these two side chains of glutamic acid and lysine because they're oppositely charged. And the third type of bonding we have is sulfur-sulfur bonds. And this only occurs when we have or when we use the amino acid cysteine. And that's because it has a side chain with this CH2SH group. And it's the sulfur that we need for these sulfur-sulfur bonds. And so what happens is that under suitable oxidizing conditions, 2 of these cysteine molecules may react together to form these sulfur-sulfur bonds. And this forms a bridge between the 2 molecules and creates a double amino acid called cysteine. And this is called either sulfur-sulfur bridging or a disulfide bridge. And so on the diagram on the right, we have 2 cysteine amino acids, and if we put them under suitable oxidizing conditions, they can form a bridge together where this section here, where we have this bond, this is the disulfide bridge. Now we're going to look at the structure of proteins. So we can actually get primary, secondary, or tertiary structures of proteins. And firstly, looking at the primary structure, this is the sequence of amino acids which have joined together to form the protein chain. So for example, we could have glyval-ala. So this is a protein chain formed from the amino acids glycine, valine, and alanine. And these amino acids are all joined together by covalent bonding. And so we need to boil the proteins for 24 hours with 6 moles per decimeters cubed of hydrochloric acid in order to hydrolyze them, which means breaking down the protein chain back into all of its constituent amino acids. And because these covalent bonds are really, really strong, and so they require a lot of energy to overcome, and that's why we need these harsh conditions. We need to boil them for 24 hours, and we also need a very strongly acidic condition. And then the secondary structure, this occurs when the chain of amino acids, so the primary structure, fold into either an alpha helix or a beta pleated sheet. So if we have a look at the diagrams on the right, here we have a beta sheet. So we have this beta pleated sheet. And here we have the alpha helix. And these structures are held together by hydrogen bonding. And this is between the NH groups and the CO group, because we know that the hydrogen is not very electronegative, but the oxygen is very electronegative. So we have a partially negative charge on this oxygen and a partially positive charge on this hydrogen. And so this oxygen and this hydrogen can hydrogen bond together. And so this is what we see here on the diagram. We've got an oxygen and a hydrogen, and an oxygen and a hydrogen, and they're all oxygens and hydrogens bonded together by hydrogen bonding. And we can use gentle heat or slight change in pH to overcome these hydrogen bonds. And this is because hydrogen bonds are a type of intermolecular force, so they don't take very much energy to break. And so that's why we only need very gentle conditions, so quite a low heat and only a slight change in pH. Whereas when we looked at the primary structure, because all the amino acids were joined together by covalent bonding, which is a very, very strong type of bonding, we need harsher conditions to break them down. And then finally, looking at the tertiary structure, this occurs when the alpha helix or the beta pleated sheet structures fold into 3D shapes. And these 3D shapes are held in place by hydrogen bonding, ionic interactions, and also sulfur-sulfur bonds, as well as van der Waals forces between the molecules. And many proteins actually fold into globulous shapes. And the shapes of protein molecules are really vital for their function. So, for example, in enzymes. So, on the right, we have this 3D globular structure. So this is our tertiary. So this is our 3D shape. We don't have a special name for it. And so we have the disulfide bond or the sulfur-sulfur bond between the two sulfurs if we've got cysteine. So this here is a cysteine. We only get sulfur bonds with cysteine because no other amino acids contain sulfur. And then we have ionic bonding between the oxygen, which is negatively charged, and this nitrogen, which is positively charged, because we have opposite charges, so we get ionic bonding or ionic interactions. And then here we have the hydrogen bond between a delta negative oxygen and a delta positive hydrogen. So now we're going to talk a bit about the bonding in wool. And this is a real life application of a secondary structure of protein. So wool is one of the naturally occurring proteins, and it's a protein fiber with an alpha helix structure. And so the secondary structure of protein. And so we know that this is held in position by hydrogen bonds. The hydrogen bonds occur between this C double bond O group and also the NH group. So we have this C double bond O and the NH of the amine. And the C double bond O comes from the carboxylic acid. So what we have is we have C double bond O and then NH, and this is attached to other things, and we have a hydrogen bond here. And so when wool is stretched, the hydrogen bonds stretch and the fibre extends. This is because hydrogen bonds are relatively strong because they're the strongest intermolecular force. And so when we gently stretch it, these hydrogen bonds will allow us to stretch. And so when we release the stretch, we allow these hydrogen bonds to return to their normal length. And so the fibre will return to its original shape. However, because hydrogen bonds aren't proper bonds, they're not very, very strong, and so they can't withstand a very high temperature. And so when we wash wool at high temperatures, we can permanently break these hydrogen bonds, and so the garment may permanently lose its shape forever. So the hydrogen bonds break, and so the garment will lose its shape. And this is because hydrogen bonds don't take very much energy to overcome. And so when we stretch them very, very gently, then they will stretch. But when we wash them at really high temperatures, then they will break. Now we're going to look at how to find the structure of proteins. So the shapes of proteins are of great importance, and determining the secondary and tertiary structures can be essential. And X-ray diffraction can locate the actual positions of atoms in space. The first step in determining the primary structure is to find out the number of each type of amino acid present in the protein. And so what we do to start is that we reflux this protein with the 6 moles per decimeters cubed of hydrochloric acid. And so what we're doing is hydrolyzing the protein. So it's equivalent to boiling the protein for 24 hours. And this breaks down the amide bond. So it breaks down the peptide link that we've seen, which is this C double bond O and then NH. So this is our amide link, or our peptide bond. And this is between all the amino acids. And so if we've broken them all down, then we get a mixture containing all the individual amino acids which were in the original protein. And so what we can do after that is use thin layer chromatography to separate the amino acids. Now we're going to talk about thin layer chromatography. So this is the method we use to separate and identify the amino acids obtained after the hydrolysis of a protein. So again, hydrolysis is where we boil a protein for 24 hours and with 6 moles per decimeters cubed of HCl. And this breaks down all the peptide links, or the amide links, the peptide bonds, to obtain a mixture of all the separate amino acids that we have in the protein. And so we then use the thin layer chromatography to separate and identify these amino acids from the mixture. And thin layer chromatography, or TLC, is very similar to paper chromatography, but instead of using paper, we're using a chromatography plate, which consists of a thin, flexible plastic sheet. And we coat it with a thin layer of silica, which is silicon dioxide, so it has the formula SiO2, and that's a solid. And so how it works is that we call silica the stationary phase, and the solvent, which moves up the plate and helps separate this amino acid mixture, is called the mobile. And the intermolecular forces between the amino acids and the solvent, or the silica, determines their affinity towards the stationary and mobile phases. And so what we mean by that is that, say the amino acid has a very strong intermolecular force with the solvent, for example. So that means that it's more attracted to the solvent. There are more intermolecular forces. And so its affinity towards the mobile phase, which is the solvent, is higher, just meaning that it's more likely to stick with this mobile phase. And where each amino acid ends up on the plate depends on its affinity for the stationary and mobile phases. So looking at the example I just talked about, if we had a higher affinity for the mobile phase, so the solvent, that means that because it sticks to the solvent more, it'll end up higher up onto the plate. So amino acids are colourless, and this is a really important thing to remember. And so the positions they have reached have to be made visible for us to be able to calculate an RF value, which we will look into a bit later. And this is done by spraying the plate either with a developing agent, such as ninhydrin, which reacts with amino acids to form a purple compound. So we'll get purple spots on the plastic sheet, or by shining ultraviolet light onto the plate. And so we have two general rules. The first rule is that the stronger the intermolecular forces between the amino acid and the solvent, the stronger its affinity for the mobile phase and the closer it is to the solvent front. So what that means is that if we've got stronger forces between the amino acid and the solvent compared to the amino acid and the silica, that means that it'll stick to the solvent more. And because the solvent is moving up the plate, that means that where the amino acid ends up is closer to what we call the solvent front. And that just means where the solvent has ended up. So the word affinity just means basically its attraction to which phase. So in this case, it's a stronger stronger affinity for the mobile phase because the solvent is what we call the mobile phase. Basically, it's the phase that always moves, compared to the silica, which is the stationary phase because it's not moving. And so the second rule we have is that the stronger the intermolecular forces between the amino acid and the silica, the stronger its affinity for the stationary phase, and the closer it will be to the origin. And the origin just means where the solvent, where the amino acids start. So how we make sense of this is that because there's stronger intermolecular forces between the amino acid and the silica compared to the amino acid and the solvent, that means it sticks to the silica more. And because the silica isn't moving, that means that where the amino acid ends up will be much closer to where it started off as, compared to where the solvent has ended up, because it's more stuck to the stationary phase, the silica, compared to the solvent. And we can use its retention factor value, or its RF value, as a measure of where the amino acid ends up on the plate. And we can compare an amino acid's RF value with known RF values from known pure amino acids, which we use the same solvent for, to identify them. And a really important thing to remember is that these known RF values are from pure amino acids, and we use the same solvent. And so if we have a quick look at the diagram on the right, so what we have here, this dotted line, this would be our origin, and the solvent front's already labeled, so that's just where the solvent ends up. And to calculate an amino acid's RF value, we do the distance moved by the spot divided by the distance moved by the solvent. So it's basically a ratio of how much the spot has moved and how much the solvent has moved. So in this example, we're told to work out the RF values of amino acids on this TLC plate. So for X, what we do is we have the RF value is equal to the distance moved by the spot over the distance moved by the solvent. So the distance moved by the spot is from the origin to the central bit of the spot. So we have that central bit of the spot is at this 0.80 centimeters. And it doesn't matter what our units are because it's just a ratio. So as long as we use the same units for the distance moved by the spot and the distance moved by the solvent, we should be fine. And so the distance moved by the spot is 0.80. And then the distance moved by the solvent, so where the solvent front is this 2.75. So all of these RF values will be over 2.75. And this gives us 0.29. And then for Y, the RF value is equal to the distance from the central part of Y, so at the center of this dot, all the way to the origin. And so that's 2.15 over 2.75. And so we get 0.78. And then for Z, we have that the RF value is equal to the central part of Z, so where this 1.70 is, and then all the way to the origin. And so we have 1.70 over 2.75, which is equal to 0.62. So now we can work out what the RF values actually mean. So we can see that spot Y has moved further up the plate compared to X and Z. And so we can say that Y has a stronger affinity, so basically a stronger attraction, stronger intermolecular forces to the moving part, so we call it the mobile phase, which is the solvent, And so then we can say that it has a higher RF value, because we can see that from all our values, because this Y here has the biggest RF value. It's probably best to say 4. So Y has a stronger affinity for the mobile phase. And so that's why it moves further up the plate. And from all the RF values, we can see that Y has the biggest RF value. And so what we can take away from this is that higher RF values means that we have a higher affinity for the mobile phase. And so the opposite is true as well. So with lower RF values, we have a lower affinity for the mobile phase or a higher affinity for the stationary phase. And this makes sense because if we think about the ratio of if we have a spot which has gone all the way to the solvent front, then we would have 2.75 over 2.75. So that would give us 1. And that's the maximum value we can have. And so it would have an 100% affinity for the mobile phase. So it has no affinity to the stationary phase because it goes all the way to where the solvent front is. Now we're going to have a look at another type of thin layer chromatography, which is 2D thin layer chromatography. So 2 amino acids often have similar RF values in a particular solvent, and so just using thin layer chromatography makes it hard to distinguish between them. And so this is where 2D thin layer chromatography comes in. So how it works is that a square piece of TLC film is used instead of the rectangle that we had for normal thin layer chromatography. And it's spotted in one corner. So we drop one amino acid, so the amino acid which has similar RF values to another one, we spot one of them in the corner. And so say we have 2 amino acids which have similar RF values from thin layer chromatography. Then what we would do would be to run 2D thin layer chromatography with both of them separately. So first we would take one of them and then spot this in one corner. And then we run the chromatogram in the usual way. So we put this film into some of the solvent and then wait for the solvent to go up the piece of plastic. And then you should have spots which have been separated. So we have spots separated along one side of the plate. So this is what we have in B. So A is where we spot an amino acid in one corner. So this spot here is our amino acid. And then for B, we run the chromatogram. So we're putting it in the solvent. And then for C, so now we have the spots separated along one side of the plate. And now we turn it around 90 degrees. And then the chromatogram is run again, but with a different solvent. And this makes it easier to see the separation between the two spots. And it also gives us 2 RF values. So after we've turned it around 90 degrees and then run the chromatogram, what we'll have will be the TLC film in F. So this is our final TLC plate. And so basically what we've done is separated spots even further. So we separated it once by with using one solvent, and then we've turned it 90 degrees and then separated those spots with a different solvent. And so this means that we have 2 RF values for this single amino acid now. And now we can match it up to databook values, so known values for pure amino acids, which have been taken in these same 2 solvents if the RF values are exactly the same.